Hi everyone, Mrs. Ortega here. I am getting ready to look at the second half of chapter 41, where we were looking at the digestive system and nutrition. So here we left off with evolutionary adaptations of a vertebrate digestive system, how it correlates with the diet. So that's where we will start right here on this slide in chapter 41, section 4. And so now we're going to take a look at adaptations to the digestive system of vertebrates. So there are quite a few. There are dental adaptations, stomach and intestinal adaptations, and mutualistic adaptations. And so these things, in general, you'll want to understand what they are and how they're adaptations with the diet. And so things as we go through here are kind of pointed out or labeled as we go already. So the first one, dental adaptations. Essentially different shapes and sizes of teeth allow for different diets to be eaten better. And so dentation is the animal's assortment of teeth. This is one example of structural variation reflecting diet. And so the success for mammals in terms of eating, capturing prey is going to be in part due to their teeth, which is specialized for different diets. And so let's take a look at some examples here in terms of some specialized teeth. Teeth of poisonous snakes can be modified as fangs for injecting venom, but other versions of teeth are shown here in this image. And they've labeled these based on their food intake, so carnivore, meat only, herbivore, plant only, omnivore, both. You'll see different combination, different amounts of teeth, different types of teeth. So the incisors and canines there are for grasping, holding onto, and ripping prey, whereas the premolars and molars are more of a grinding motion for plant-based foods. And so as you can see, in omnivores, you have a good combination of both. Carnivores, their incisors and canines are quite large, as well as their premolars or molars are more jagged to modify for their food to tear apart that really tough meat, whereas the herbivore has very reduced incisors and canines and a lot of premolars and molars to allow for that chewing of those plant material. So dental adaptations, you want to understand what this is different types of variations of teeth that allow for eating different diets. Now on to stomach and intestinal adaptations. Those are different variation of the alimentary canal. So remember, the alimentary canal is the digestive system canal starting with the mouth through the passageway of different organs all the way to the anus. So that's the canal itself. And so there are variations of this canal based on the food intake. So herbivore, omnivore, and carnivore all have different alimentary canals, usually defined by species. And so let's take a look at two examples here in this next slide. The first one is the coyote, and the second one's the koala here. You can see their alimentary canal starting from the stomach all the way to the large intestine. The small intestine of the carnivore is much longer, and that is to absorb, break down, and then absorb those nutrients. And so breaking it down takes a lot more effort than the herbivore in terms of the nutrients that are being absorbed from the type of food that's being eaten. Whereas the large intestine is rather short in the carnivore to absorb water, the functioning, and other vitamins in the large intestine. For the koala here, the herbivore, you can see starting from the stomach, the small intestine is not quite as long as the carnivore, but they do have a very large cecum that you can see there for it houses bacteria and other small microorganisms that will digest the plant material. The tough material like cellulose and lignin can be broken down there, and so then it can be absorbed by the animal. And so here you can see they do have a very long, large intestine, colon region to absorb as much water as they can, and possibly in some species other vitamins and minerals, or vitamins and things like folic acid. And so the alimentary canal is adapted for different types of food, and so that is a key trait here you'll want to understand. Lastly, mutualistic adaptations. Mutualistic adaptations sounds like its name, in that mutualism is the relationship between two organisms where they're both benefited. So both the host and the organism living within the host are benefiting from the relationship. And so here, this is for humans. The coexistence of humans with bacteria is a mutualistic symbiosis for the most part. Any non-mutualistic symbiosis is going to be a bacterial infection that's going to cause some type of disease, like a respiratory infection, sinus infection, some other infection. And so the bacteria that lives mutualistically in humans will mostly be in the intestine. And so in that area, they will actually produce vitamins. The bacteria will use the food that's being ingested and produce vitamins from it that we can then absorb. Some examples are vitamin K, biotin, folic acid. The bacteria is also regulating the development of our intestinal epithelium and helps with some innate immune system functions. And just some side facts here, that there is anywhere from 10 to 100 trillion bacteria living in the human digestive system at one point. And one strain, E. coli, is so common and in large amount that it is used, the detection of E. coli is used to detect human contamination in water, like lakes, ponds, streams. Detecting for E. coli can help actually detect for human waste. So this next section here looks at this a little bit more and goes into the microbiome. So the microbiome is the collection of microorganisms living on the human body. And so this is a new area of research, a more recent area of research that they're using the polymerase chain reaction, or PCR, to study the microbiome. And so there are differences based on diet, diseases, and age. Another thing about bacteria and mutualistic adaptations, one bacteria called H. pylori is known to disrupt stomach health by eliminating other bacterial species from the stomach, and also interrupting the epithelial layer and mucosal layer of the stomach. And so to understand the microbiome, here is a diagram of a variation of the gut microbiome at different life stages, and these are different phylum of different bacteria. And so by examining these and comparing these, we can understand more about our health, we can understand more about digestion and the nutrients that we're absorbing in our bodies, as well as maybe possible treatments for diseases in terms of digestive system related diseases. Here we can see the stomach microbiome, again, but with the H. pylori infection. This actually causes those stomach ulcers, once believed to be Stomach ulcers were once believed to be a result of a low pH inside the stomach. It is now proven that it's actually due to the presence of H. pylori causes stomach ulcers and lesions in the stomach, and can be very, very detrimental. And so treating it with a specific antibiotic for H. pylori will cure the stomach ulcer and resolve all symptoms. But to test this, two scientists actually decided to ingest a contaminated milk solution with H. pylori in it, and then subsequently got the stomach ulcer symptoms and H. pylori infection. They won the Nobel Prize in 2005 for this discovery, but it was definitely not the approved method of testing. So, interesting note there. Mutualistic adaptations in herbivores are much different. They have much different structure in the fermentation chambers where they digest cellulose from plants. These particular examples are not anything you have to memorize, but it is very interesting. For rabbits and rodents, they actually will pass their food through their alimentary canal twice. So after the first time they ingest their food, get the excrement, they will actually re-ingest the excrement. And this process is called coprophagy. It's actually very well studied to understand more about why they do these things. And so they actually get more vitamins out of the feces that they ingest. Let's see here. Giant tube worms are an example. They don't have a digestive system, instead obtain nutrients from bacteria within their bodies. So there are many examples of mutualistic adaptations in herbivores. One example for animals is the ruminant example, where they have very elaborate herbivorous diet adaptations. So here you can see ruminant digestion in a cow. They have different chambers for different stages of the digestion, the fermentation of the plants to break down the grass here to get the nutrients. And so now that we've looked at adaptations for the digestive system, we'll take a look at regulation. So we'll see how the digestive system is regulated, along with glucose, sugar, energy levels within the body. And so these processes have to match the organism's circumstances and the need for energy. So first we'll look at regulation of digestion. So each step in the digestive system is activated as needed. So that starts off with the mouth. The oral cavity will secrete saliva in the presence or detection of food. So each step has a way of activation. And so the enteric division of the nervous system helps with this digestive process, as well as the endocrine system. And so in this section, we'll look at the endocrine system and how it regulates digestion through the release and transport of hormones. So this first example here is going to be after food has been ingested, the food passes through the esophagus to the stomach. So the food entering the stomach will stretch the stomach walls. So this will release gastrin hormone. This gastrin hormone will then go to the capillaries in the blood vessels and will circulate through the whole circulatory system back to the stomach, where then gastrin will then bind to the cell receptors and stimulate gastric juice being produced. And so this is a stimulatory pathway. So it's highlighted in green that gastrin is the hormone release that will stimulate gastric juice forming. Another example here, another hormone control, is when the now digested food, the chyme, is going to enter the first section of the small intestine, the duodenum. It's going to release hormones called CCK and secretin. CCK stands for cholecystokinin. It's written down here for you. I'm sorry, I cannot pronounce that one very well. And so that is going to stimulate the release of the digestive enzymes from the pancreas and the bile from the gallbladder. And so that is CCK. Secretin will stimulate the release of bicarbonate, HCO3- from the pancreas into the small intestine. And so both of these are stimulatory hormones that will go to the small intestine after entering the circulatory system. So they're released, they're enter the circulatory system. For CCK, it will go to the gallbladder, it will go to the pancreas, and then bind to those cells and trigger the release of enzymes and the release of bile. For secretin, it will go to the pancreas and trigger the release of bicarbonate, HCO3-, and those will then go to the small intestine through the little tube there. And then the third regulation shown here is actually the only inhibitory pathway that we'll discuss, and that is when the levels of secretin and CCK are too high in that region that we just saw, that this will actually inhibit or slow down the movement of the stomach, so the peristalsis of the stomach, and inhibit or stop gastric juice secretion. So this is going to slow the digestion process, and so that gives it more time to digest the chyme that's already entered the small intestine. So when it's really rich in fat, it's going to take more time to break that apart. And so it's messaging the stomach and saying, hey, slow down, I need more time to break this apart. And so those are the hormones and how they play roles in digestion. You'll want to know those, understand at what level and what level of hormone is, and then what it does. So if it stimulates digestion, if it inhibits digestion, where it's secreted, and where it goes to. Now we'll take a look at regulation of energy storage. So this will be the aspect of glucose and regulating glucose. So for humans, energy is stored first in the liver, then the muscle cells as the polymer glycogen. So this is monomers of glucose linked together. Then after that, excess energy will be stored as fat in adipose cells. So then when fewer calories are taken in, then more than when they're taken in less than expected, the human body will expend first the liver glycogen. So it's easiest to access is the liver glycogen. We'll break it down to glucose. Then it'll use the muscle glycogen, and then lastly it'll use fat from fat adipose cells. And so that's why it's very difficult to lose fat, as you have to first take in fewer calories that are needed. Then you have to expend the glycogen from the liver and muscles, and then you can start burning fat. So it's very, very difficult process. And so levels of glucose need to be maintained in the body. These are done with regulation done by hormones insulin and glucagon. So they will regulate the breakdown of glycogen into glucose and the building of glucose into glycogen. The liver is a site for this process of regulating the levels. So we have the description here. But we also have an image right here that kind of describes this section. I like to look at the image as we go through it. That the normal blood glucose levels are anywhere from 70 to 110 milligrams of glucose per 100 milliliter. If you get a test done, a blood sugar test, you'll see at fasting the goal levels are to be 100 or below for most people in our age range, from I believe 20 to 30. And so that is where you want to be at fasting. When you're not fasting, it will probably be higher. It's something that you would want to measure. If you're wanting to monitor glucose levels, you want to measure it at least once a day. And so it does vary from person to person. Some people function just fine at higher levels, while others don't. So it's a unique factor varying from person to person. But in a situation where the blood glucose level rises, like after eating, the insulin will be secreted, and this is done by the beta cells of the pancreas. So the beta cells release that insulin, it circulates through the circulatory system, and then it will transport glucose into body cells and storage glucose as glycogen in body cells in the liver. And so that insulin binding to those cells says, hey, take up glucose, bring it into the cell, and so that's what the body cells and liver does, and so then the blood glucose level falls back to normal. And if the opposite happens, if the blood glucose levels fall, such after fasting for long periods of time, overnight, if sleeping through the night, waking up in the morning, your blood glucose levels will be low, and so the glucagon will be secreted by the alpha cell of the pancreas, and so that glucagon will then circulate through the circulatory system, go to the liver, and from the liver it will start releasing glucose into the blood through breaking down that glycogen, and then the blood glucose level rises. And so if the blood glucose level is continually low, then after the glycogen in the liver has been broken down, it will go to the muscle cells, break down its glycogen, and then lastly go to the fat cells. And so that's why it can be very hard to get rid of excess fat for humans. So going back to this slide just to review a bit here, insulin acts on nearly all body cells. So that will take in glucose from the blood to the cells, whereas glucagon is going to go to certain cells, cells like the liver, the muscles, and then lastly the fat cells. And so both of these hormones are made in the pancreas and the islets of the pancreas. These are actually clusters of endocrine cells, so that makes sense. These endocrine cells are known for making, producing, releasing hormones, and so there are clusters of these cells in the pancreas. So the alpha cells in the pancreas will make glucagon, and the beta cells in the pancreas will make insulin, as we saw in that diagram there. So note, the only cells or exception to this regulation will be brain cells, as they need to be able to take in glucose whenever they need it, whenever glucose levels become low. If they're burning more energy, then they're going to take in more glucose, whether or not that insulin is present in the circulatory system. Alrighty. So we have a video about regulating blood sugar that we'll see here in just a minute. It also does tie into diabetes. So diabetes is a very common disease in the United States as well as worldwide. So this is caused by a deficiency of insulin or decreased response to insulin in target tissue. And so when the response or the deficiency of insulin is present, then cells are unable to take up that glucose to meet the metabolic needs. And so the level of glucose is usually high in the blood, and it may exceed the capacity of the kidneys to reabsorb it, and so therefore then some of the sugar gets excreted in urine. And so that is one test for diabetes, is finding sugar in the urine. Usually it is a sign of diabetes. Type 1 and type 2 diabetes are very well studied and well known. Type 1 is an autoimmune disorder in which the immune system destroys the beta cells of the pancreas. This usually shows up during childhood and has to be regulated for the lifetime of the adult. Thankfully, now we have a lot of different options in terms of regulation with a continuous blood glucose monitoring system from Freestyle and some other manufacturers. They can continually at any time record their blood glucose level with that patch inserted onto their arm. So it makes it very convenient. As well as many of these are available for anyone with diabetes. Continuing on to our PowerPoint here, type 2 diabetes is characterized by the failure of the target cells to respond to insulin. So the insulin is being produced, is being secreted, but the response isn't there. So the cells don't take in the glucose like they should. And so this is known to be caused by excess body weight, lack of exercise, usually appears in people over the age of 40, but can develop in younger people. There are other regulations of appetite and consumption that is discussed here, in that overnourishment causes obesity, which is excessive intake of food, which is stored as fat. Obesity contributes to a lot of different factors, different illnesses like the diabetes, colon cancer, breast cancer, heart attacks, stroke, all can be correlated with obesity in some form or fashion. And so researchers have been studying obesity and other illnesses related to this and have discovered many mechanisms that help regulate body weight. And some of them are shown here. That these hormones regulating long-term and short-term appetite affect a satiety center in the brain. The center of the brain that says if you're hungry or not can be regulated by hormones. Some of these hormones are shown here. Ghrelin is a hormone that is secreted by the stomach wall that triggers feelings of hunger. Insulin and PYY is hormone secreted by the small intestine after meals. Both suppress appetite. And then leptin, one that's been studied well for obesity, has been produced by the fat tissue, also suppresses appetite. And so malfunction of hormone leptin, while very rare, can lead to obesity. So there's a lot of study out there about hormones, how they affect appetite and consumption, and how we can regulate and control the obesity issue that is worldwide, but is especially in the United States. So this wraps up this section on Chapter 41 in Nutrition Digestion. I hope that you find this to be informative and helpful. If you have questions, please let me know. Next video, we will look at Chapter 42. Bye-bye for now.